The first stage of cell division is interphase, and the first stage of interphase is growth and preparation. To begin with, the cell is growing. It needs to make proteins essential for cell function. The DNA molecule inside the cell is made up of 4 nitrogen bases: adenine, thymine, cytosine, and guanine. Adenine always pairing with thymine, and cytosine always pairing with guanine. The sides of the DNA molecule are made of sugar and phosphate. To make a protein, the DNA unzips. Free-floating nitrogen bases come and attach themselves to the DNA molecule. Then, after making a copy, and it is important to note that there is no thymine in RNA. There is uracil,
which is an amino acid.
The nitrogen bases come alongside the ribosome and get fed through it to make the protein. The DNA rezips and is back to normal. The second stage of interphase is replication. The cell unzips once again and nitrogen bases come again and attach themselves to the DNA molecule. This time they make a perfect copy. This is important because the two cells that are going to become because of the division need to be identical. There are now 2 identical copies of DNA. This allows the cells to be identical. The cell rezips. The third stage of interphase is continued growth. DNA is in the form of chromatin, which is coiled up chromosome. This is important because RNA still needs to be made so that proteins can be produced. It needs to be loosely coiled so that sections of it can unwind for the RNA to be made. Organelles will need to be duplicated in this stage for the new cell.
To start prophase, chromatin becomes X-shaped chromosomes and become visible under a light microscope. What you will see happen is the nucleus will disappear and the membrane will start to break down. Also, spindle fibers begin to form and stretch across the cell from the centrioles.
This is late prophase. The spindle fibers complete forming. The chromosomes attach to the spindle fibers at their centromeres, which is here. And the nuclear membrane, as you saw in the other video, around, has disappeared. The tugging action of the spindle fibers pull the chromosomes into the equator or middle of the cell. This is anaphase. The spindle fibers here begin to shorten and contract, thusly pulling these chromatin in half. And again. Yeah, okay, and one more. And they have pulled apart. They come in to the end here.
Okay. Now, telophase. As you can see, one complete set of chromosomes is in each half of the cell, and the nuclear membranes have reformed. Also, what will happen in this phase is the spindle fibers will begin to disappear. And also what will happen is both the 2 nucleoluses will come back and be back in the nucleus.
The final stage of cell division is cytophesis. During this, the cell separates into 2 identical cells. How? The cell membrane pinches together and we are left with 2 identical cells.